Okay, this chapter covers T cell activation. So far, we've covered antigen presentation to T cells. After this, we're going to talk about the differentiation and functions of both CD4 plus and CD8 plus cells. So that's going to kind of cap off the T cell chapters. Then we're going to get into B cell activation and functions of the humoral immunity. After that, we get into antigen tolerance, autoimmune diseases, cancer pathology, and all the cool ways biomedical engineers use the immune system to help fight these things. But back to T cell activation. First, I want to give a one slide recap of everything that we've learned so far about activation, as also kind of an introduction to this particular chapter. So, T cells, we know, are generated in the central or the generative lymphoid organs, but they never get activated there. They never see an antigen, something that's foreign to the host body, in the generative or central peripheral organs. They only encounter antigens and get activated in the secondary lymphoid organs. This is primarily the spleen and the lymph nodes, but could also occur in the mucosal lymphoid tissues. We know that in 99% of the cases, it's dendritic cells that act as the activating antigen presenting cells, and dendritic cells concentrate in areas of high T cell concentrations. And this is where they simultaneously present multiple antigens. And I want to point out that the dendritic cells never know what antigen is a magical antigen that they're supposed to present. So they don't just present a single antigen. They digest and chew up many different types of proteins and display all kinds of peptides, even host peptides, self antigens, at all times, just hoping that one of them is going to link up with the T cell. We also know that once activated, the fully differentiated effector cells can go to the sites of inflammation or infection and further respond to antigen and stimulation by antigen presenting cells there. Sometimes confusingly, I think the book uses activation in this case as well, so I'm going to be trying to be really careful in this particular chapter not to use activation as just a catch-all word. So for the purposes of this chapter, activation is the process of going from naive to differentiated and activated effector T cells, and then those T cells can further respond to antigen in order to carry out their effector functions. So it's always continuously necessary that they respond to antigens in order to perform their jobs. We've also covered a few of the biological responses that rise about through activation. So we know that activated T cells secrete certain cytokines that perform various functions, such as they promote antigen presenting cells' capabilities. As I just mentioned, since effector cells need to continuously respond to antigens, it's important that after that very first and only single dendritic cell that was displaying that antigen that caused the clonal expansion of the T cells, that the T cells promote other antigen presenting cells to continuously display those antigens and carry out their functions. Okay, so cytokines, for instance, will cause an upregulation of the MHC complexes that help display those antigens. Another reason why this is important is that APCs not only display antigens, but they also provide stimuli that guide the magnitude and nature of the T cell response. There's even surface molecules on APCs that inhibit activity, right? This is sort of a form of a regulatory negative feedback system. We learned that the activation of naive T cells induces the rapid proliferation of them. This is a process known as clonal expansion, where copies of the single T cell clone that's specific to that antigen makes many, many copies of each other in order to produce that arsenal, that army of T cells that can then fight off that potential infection. During clonal expansion, it's not just one type of T cell that's developed, but many different subtypes. Most importantly, both effector T cells of both the CD4 and CD8 plus variety, and also memory cells that don't have effector functions, that is, they're not involved in killing infections or microbes or getting rid of antigens. Instead, they're meant to hang around much long after the infection or antigen is gone and wait for secondary or tertiary responses, where they're much more effective than a naive T cell would be. The memory cells generated here can be part of both a circulating pool, and they also reside in peripheral lymphoid organs, primarily mucosal tissues, where a T cell might first encounter a pathogen more directly as opposed to waiting in the lymph nodes. And lastly, in the later stages of infection, after most of the antigen is gone, effector cell population starts to decrease and eventually dies off entirely. And this is due primarily to the fact that T cells or effector T cells need antigen stimulation as part of their survival mechanisms. Without antigens, they will die absolutely. Now, there's a few things that T cell must recognize in order to become activated. The antigen is the primary signal, and it's what provides and ensures the specificity of a response. Okay, the T cell receptor is, just like antibodies, highly specific. However, that alone can ensure activation. T cells also require a combination of co-receptors, co-stimulators, and cytokines, typically originating from other APCs or cells of the innate immune response. This helps ensure that the T cells recognize that there is truly a danger present and that it's not detecting, say, something like a self-antigen. So there are only signals present during the inflammatory or infection process that help activate a T cell. Now, to activate naive T cells, dendritic cells, I keep saying, are the primary antigen-presenting cells in order to do this. There's a few reasons for that. There's some minor reasons for the fact that they display the most and diverse array of pathogen-associated molecular pattern recognition receptors, like toll-like receptors. But the primary reason is just due to their migratory habits and their ability to go from the site of an infection, process the antigens, and then migrate to the peripheral lymphoid organs where they can concentrate in areas of high amounts of T cells. And it's due primarily to the chemokine receptor CCR7. That's one I keep mentioning in many lectures. That's the one I remember through Creedence Clearwater Revival, one of my favorite bands, and then the soccer player Cristiano Ronaldo, who wears the number 7 jersey. So CCR7. And then you just have to worry that the weird ligands that attach to it, they don't follow the same number. There's two ligands. It's CCL19 and CCL21. Again, the CC always has to be the same for a ligand receptor combination. You know that L is the ligand, R is the receptor. So all you have to remember are odd numbers. That's how I remember it. 7, 19, 21. But to bring it back to the migratory patterns, both T cells and dendritic cells have these same chemokines that respond to them in the same way, and that's why they end up in the same T cell zones in the lymphoid organs, in the lymph nodes, to be brought together. And it's all because of dendritic cells that these antigens are first brought from either the epithelial barriers, or in the case of a bloodborne pathogen, it'll be brought to the spleen, where there's T cell zones as well. But that's why it all gets initiated and why it happens initially. It's due to dendritic cells. And this is another recap slide from a previous chapter. I think this is chapter 6. But once activated, differentiated effector T cells, like CD4 and CD8 plus cells, can further respond to other antigen-presenting cells. So in this slide, I already have dendritic cells activating those naive T cells, become effector T cells. Macrophages can present antigens and receive signals from CD4 plus cells. B cells can also present antigens and receive signals from CD4 plus cells. And then remember, any cell that can get infected by viruses or become cancerous can present antigens to CD8 plus cells. They do these through either class 2 MHCs, and I said class 2 because those are the ones that present antigens to CD4 plus cells, and then class 1 MHCs to CD8 plus cells. Again, the way I remember that is that the lowest and the highest number kind of bookend the pairings, so 1 goes with 8, and then in the middle, you have the two middle numbers. 2 goes with 4. So 2 slides ago, I just mentioned the concept of signal 1, signal 2, and the necessity of co-receptors and co-stimulators. So we're going to talk about that over the next few slides. So as I mentioned, in order to get activated, in addition to that antigen T cell MHC complex, T cells also need signals through co-stimulators that act alongside antigen presentation in order to activate the T cell. So in this diagram, we have a very famous combination that we're going to talk about pretty soon, the B7 and CD28 co-stimulators that helps activate T cells, activate T cells know then to secrete cytokines and tell other T cells that, hey, we got a job to do. In the absence of an inflammatory state or an infection where antigen-presenting cells are, we'll say, in the unactivated state, where they lack these co-stimulators because they haven't been given signals otherwise, they can potentially still attach to naive T cells, but the T cells will fail to activate, where they might activate, or actually they might already have been activated. So if the antigen is still there, but the other co-stimulators aren't there. But they might enter a state of prolonged unresponsiveness, something that we call peripheral tolerance. And it's a subject of one of the later chapters in the book. The net response is that these T cells become ineffective and unable to carry out their functions. Now, I have here a slide that I brought up, I think, in week 2 or 3 of the IG superfamily when we first started to talk about both antibodies. We're not going to talk about the T cell receptors in depth in this class. We certainly talked about class 1 MHCs. ICAM1 is an adhesion molecule that actually has a large role in activation of T cells as well. They have roles in the co-receptors of T cells. But now we're going to get into CD28. We just saw an illusion of that in one of the previous pictures of B7 binding to CD28. And it's one of the co-stimulators that we're now going to get in more detail. Now, CD28 is one of the best studied co-stimulator receptors, and it binds to the ligands in the B7 family. It used to be thought that B7 was a single ligand. We now know that there are two very structurally similar ligands called B7-1 and B7-2. Now, the first thing I want you to take note of is where CD28 is and where the B7 ligands are. CD28 is a receptor, so I like to think that it has downstream signaling processes like receptors do. Ligands tend to not do that. So the T cell is what needs the second signal in order for activation. So it's the one that has the receptor. So CD28 is always on the lymphocyte, on the T cell, whereas B7 is always on the antigen-presenting cell. Okay, antigen-presenting cells are told through cytokines during the innate or inflammatory response that, hey, there's danger, you should express B7 in case you find a T cell. So they express them. They don't respond to the binding of B7 by T cells. It's only the T cell that really responds to the signals, okay? So they have the receptor, antigen-presenting cells have the ligand. Okay, I just mentioned that ligand expression is induced by the innate immune responses, so stimulation by toll-like receptors, IFN gamma, CD4 plus cells also help promote this. It's for a form of positive feedback. So in short, the expression of B7 fluctuates according to the response that the body needs, according to the innate immune response. CD28, on the other hand, is expressed constitutively, so its concentrations never change on naive T cells. Now, if you had to guess which antigen-presenting cell typically expresses the highest amount of B7s, if you got this wrong, you probably haven't been paying attention throughout the entire lecture, but if you had to guess, it's got to be dendritic cells, and you would be absolutely correct. It's one of the many reasons that makes them one of the most effective antigen-presenting cells. We learned previously that adjuvants are a component that help out an essential role in inducing primary T cell responses to protein antigens in vaccines. Well, here, now we're starting to understand what adjuvants do. And adjuvants are used to help induce high expressions of co-stimulators like B7. And it's important here to note that adjuvants are not co-stimulators themselves, but they induce the expression of co-stimulators. The CD28-B7 interaction also works to cooperatively promote survival, proliferation, and differentiation of T cells. As I said earlier, that T cells require antigen stimulation in order to survive. It's also in cooperation with CD28 and B7, as this stimulation increases the expression of anti-apoptosis proteins. I've labeled them here BCL2 and BCL-XL. We learn about them in later chapters, but just know that this is one of the mechanisms that helps them stay alive. Now, activation from naive T cells to effector cells requires a large amount of these CD28 B7 pairings. It's important to know that once activated, effector T cells still need this expression, but to a lesser degree, and memory T cells even to a lesser extent. So memory T cells have already previously been activated, as are effector T cells. Once they find their antigen, they already know they have a job to do, and they need less coaxing from CD28 and B7, although they do need a little amount. Now there's a few other co-stimulators that I'm going to mention. There's a nice little chart here on the left that helps outline it. Don't worry about that sort of grayed out area. I'm going to get to that in just a second. But the first class of co-stimulators I want to discuss are ICOS, which stands for Inducible Co-Stimulator CD278, and then its ligand ICOS-L. And you don't have to recognize these cluster names, but ICOS and its ligand ICOS-L, which stands for ligand. It's also well characterized. ICOS will come into play during T cell dependent antibody responses. We get into that, I think it's chapter 13. But ICOS-L is expressed on B cells, dendritic cells, and other cells. But for now, kind of think about that B cell and then the T cell dependent antibody response that's going to come up a little bit later. There's also a few co-stimulators under what we call the tumor necrosis factor receptor family, TNF-R, and integrins that plays co-stimulators. So CD40L, which is, as it says, a ligand, is expressed on activated T cells, and it helps stimulates CD40, which is part of the receptor family, on antigen-presenting cells to help express B7 and secrete IL-12, which both promote T cell differentiation and activation, which seems kind of like a roundabout process. So let me go in a little bit more detail on that on the next slide. So here's a situation in which CD40 and CD40L plays a vital role. So in this case, we have a dendritic cell. It's displaying its antigen. It has CD40 receptor. That's part of the tumor necrosis factor receptor family, but no B7. T cells, thankfully, express CD40L, the ligand that's for CD40. And whenever antigen-presenting cells recognize this, antigen-presenting cells recognize that CD40, the receptor, is bound to something. So they do two things. They start expressing IL-12, which helps T cell activation and differentiation. And they also express the B7 ligand, which again attaches to the co-stimulator CD28 to also induce activation. This phenomenon is sometimes called licensing because already activated T cells help license APCs to become more powerful stimulators of immune responses. Now, going back to this nice little chart of co-stimulators and co-inhibitors, I want to talk about co-inhibitors. Okay, and the first thing I want to stress is that the outcome of T cell activation is influenced by the balance of stimulating and inhibitory receptors of the generic CD28 family, which all of these fall under. So let's not think of this as, okay, are there co-stimulators? Then it's going to activate. Or are there co-inhibitors? Then it's not. They're always present at almost all times, just in low concentrations or in high concentrations. And it's the ratio or the balance of these two that determines the ultimate fate of the T cells. Now, the two big inhibitors are CTLA4 and PD-1. They're receptors on T cells that both function to limit immune responses. CTLA4, I think, is pretty accessible to us right now because it's very much like CD28. It just has the opposite effect. And I say it's like CD28 because it actually binds to the same exact ligands, B7-1 and B7-2. So if it outcompetes and if it's present at higher concentrations than CD28, it's going to bind to more of the molecules for B7 as opposed to CD28. So the more that CTLA4 binds, the more that's going to inhibit the signaling of that sort of stimulating or activation factor. PD-1 also delivers an inhibitory signal. It binds to, and this is easy to remember, the PD-L1 ligand and the PD-L2 ligand. And this actually functions by blocking downstream signaling of CD28 and B7. So B7 can still be present and be binding to CD28, but if PD-1 is activated, it blocks that signal from happening. Both of these co-inhibitors are involved in the phenomenon known as tolerance, which is where a T cell might recognize its antigen, but because it has these blocking mechanisms, it kind of thinks, NBD, and it doesn't do anything. And it gets tolerant towards that particular antigen. Now, I'm fully aware at this point that I've thrown out a lot of acronyms. These are a lot of letters that don't really spell out anything meaningful. But I promise you that these co-inhibitors are ones that you're going to be hearing about a lot in various chapters outside of this class. They're part of a new class of blockbuster drugs that companies like Merck and Bristol Myers Squibb and GSK are all about. So I want to talk about that a little bit more. So there's a few ways that you can use these co-inhibitors in therapeutic applications. There might be cases where you 100% you want to inhibit the actions of the immune system, like in the cases where you have autoimmune diseases, or in this case, rheumatoid arthritis. So in the first example, engineers have made a fusion protein of CTLA4 and then the FC portion of IgG. So in this case, CTLA4 has a higher affinity to B7 than the CD28 on the T cells, so it naturally binds to the antigen-presenting cells, preventing T cells from being activated and carrying out their effector functions. Now, you might also want to induce the exact opposite, where you want T cells to carry out their effector functions and not be inhibited. In this case, we have developed antibodies specifically for CTLA4 and PD-1 that have a high affinity towards those receptors and prevent them from binding to their respective ligands. In this cases, those T cells that are activated never become inhibited and they keep carrying out their effector functions for longer periods of time. And this is particularly important in cancer. So Merck's flagship drug, Keytruda, is an anti-PD-1 antibody. The first round of anti-CTLA4s, I think, were famously used on Jimmy Carter when he had cancer, and I think this is 2014. But this is a new class of cancer therapeutics because, as we're going to learn, T cells are involved in fighting off cancer. So this is one case where you want to stop the inhibition of T cells. Now we'll cover some of the functional responses to activation. Okay, so there's a few things that increase in surface expression in T cells once or as they're being activated. We previously, during the migratory patterns of lymphocytes, there's a protein CD69 that gets expressed on T cells that competitively binds to the S1P-R1 receptor. So S1P-R1 responds to the ligand or the protein in the circulatory system, S1P, and whenever cells express that, they want to go back in circulation. So one way of keeping cells during the activation process within lymph nodes is to express a ligand, CD69, that competitively binds to this receptor and not allowing S1P to bind to it. So in this case, T cells don't know that they want to go back in circulation. They remain in the lymph nodes. That way they can continue clonally expanding and differentiating. So first, this increases. Finally, when they want to get released from the lymph nodes, CD69 gets downregulated and gets away from the surface. Second, there's an important surface receptor that binds to a growth factor, IL-2. What's funny is that this receptor comes in 3 parts, or I think 2 parts, but only one part gets upregulated or downregulated. So the IL-2 ligand binds to the IL-2 receptor. It's the IL-2R alpha that gets upregulated during activation. So this in parts only allows activated T cells to respond to the growth factor and not other naive T cells that have yet to get activated. As discussed before, we talked about CD40 and CD40L. Lymphocytes in this case have the ligand, and it's this ligand that's recognized by macrophages and B cells to help the lymphocytes, in this case CD4 plus T cells, to carry out their functions. And then also there's CTLA4, an inhibitory receptor that's necessary for inhibition and antigen tolerance. This also gets increase in expression. In addition to that, there's also adhesion molecules, chemokine receptors, things to help the T cells navigate to their destined locations. So next up, we're going to talk about cytokines that are involved in T cell activation and functionality. For the most part, it's CD4 plus helper T cells that make up the largest amount and variety of these cytokines, in part because they're more messengers that help instruct other cells what to do. On the other hand, CD8 plus cells might respond to some cytokines, but they just have their own functions to carry out and are less involved in working with other cells. Another interesting thing to note is that most cytokines act what's called autocrine action, which means that it's the cells that produce the cytokines that want to respond to those cytokines, as opposed to paracrine action, where one cell produces a cytokine for the purpose of another cell to respond to it. Now, one of the main cytokines I want to talk about is IL-2 because of its heavy influence during the activation and differentiation function of T cell. It's a growth factor, a survival factor. It's important for the differentiation of various effector T cells, and it responds to the IL-2R receptor. So this is one of those convenient cytokine receptor combinations that have the same number. And this receptor consists of 3 non-covalently linked chains. And what you need to remember is that the IL-2R alpha, which I just mentioned on the previous slide, is the only one unique to IL-2. The other chains, in fact, are shared with a couple other receptors for different cytokines, like IL-4, 7, 9, 15, and 21, but we don't need to go into that right now. Now, it's the arrangement of these chains and the amount of the alpha portion of the receptor that determines the activation and affinity to IL-2. So without the alpha chain, like what naive T cells would have, they bind to it with a relatively, high dissociation constant, which means low affinity. So in this case, I've set a -9, and becomes a change in 3 orders of magnitude, so an increase in affinity. And then again, you have to remember with dissociation constants that they go in the opposite direction. They're inversely proportional. So it has a much lower dissociation constant, which means a higher affinity. Again, 3 orders of magnitude with that alpha chain. And it's because of the combination, but that both IL-2 becomes increased in expression during activation, and then the IL-2R alpha gets increased during activation, that ensures that during this positive feedback loop, that only T cells with that combination, with the alpha chain in the receptor, can respond to this. All right. Otherwise, other adjacent T cells could potentially react to IL-2, but since they don't have that alpha chain of the receptor, they see that cytokine around them, but they don't sense it, they don't respond to it, and it's only the clonally distributed T cells that are being activated that respond to it and continue to grow. One little cool note I found out about this particular receptor is that during the chronic stimulation of these T cells, like when there's a really strong immune response, like you might find during an organ transplant where the body is continuously trying to reject this particular organ, these T cells begin to shed the alpha chain of this receptor. In fact, you can do a blood assay that measures for the serum concentration of just this alpha portion of the receptor as a quantitative measure of a strong energetic stimulation. Now that we've discussed the receptor, I want to talk about some of the functions of IL-2 and also some of the reasons why it used to be called T cell growth factor. Okay, first and foremost, IL-2 stimulates the survival of T cells by inducing anti-apoptotic proteins like BCL2. So that's, I think, the second or third time I mentioned that protein. It also increases production of the effector cytokines like IFN gamma and IL-4 by T cells. The cytokine is vitally important for the survival of regulatory T cells. Now, regulatory T cells, as I think I just mentioned on the previous slide, always express the full IL-2 receptor, meaning they always constitutively express the alpha portion of that receptor. It never has to be upregulated for regulatory T cells. Also important to know is that regulatory T cells, unlike other types of T cells, never themselves produce IL-2. So they're dependent for their survival on other T cells in order to induce the expression of IL-2. Okay, so they're really good at picking it up because they also have the full receptor, but they don't themselves express it. In fact, scientists have carried out studies in knockout mice that have the IL-2 gene knocked out that find out that they tend to suffer from overproliferation of T cells and B cells and also high amounts of autoimmune diseases, in part because they lack the regulatory cells necessary to keep their T lymphocytes in check. And it also seems to indicate that there's other growth factors that can supplant IL-2 that can still maintain activation and expansion of lymphocytes. So it really seems more important to us from a regulatory standpoint than perhaps from a growth standpoint. IL-2, because it's a growth factor, is also really important during the clonal expansion process. So before clonal expansion, there might be one lymphocyte that's specific to an antigen for every 10,000, 100,000, 1 million, or even less lymphocytes in general. After expansion, and all because of IL-2, we see a 1,000-fold increase in CD4 plus cells and a 50,000-fold increase in CD8 plus cells. And the difference of those numbers in part due because CD8 plus cells are necessary for themselves to carry out the effector functions. CD4 plus, again, are helper T cells that help other cells carry out their functions. So they can help multiple cells do their job. CD8 plus cells have to do it themselves, which requires larger numbers. Now, lastly, I want to talk about the development of memory T cells. So memory T cells are really the heart of any vaccination program because it's less about the initial response that we care about and more about the development of memory and the ability to fight secondary and tertiary responses that we care about. And it's all due to memory T cells. So these are memory immune cells that are specific to any antigen that we've had previous exposure to, and they persist for years, decades, or even lifetime of a host that has those cells. Memory T cells can also be developed through different pathways, right? So there's what's called the linear pathway, where naive cells have become effector cells, they finish their job, and then they turn into memory T cells instead of dying off. And then there's also the alternative pathways where naive cells become activated, and during the expansion process, both effector cells and memory cells are produced concurrently. Part of the reason that memory T cells can live so much longer than other types of lymphocytes is that they have increased expressions of anti-apoptotic proteins like BCL2, BCL-XL. So I mentioned those a few times at this point. And it's because of these anti-apoptotic proteins that results in an increased lifetime for them. Paul, the hallmark of secondary and tertiary responses and the ability to have memory of lymphocytes is how much faster that they respond in subsequent reactions or recognition of those same antigens that they're specific for. So for instance, naive cells turn into effector cells on the order of 5 to 7 days. So that's how long it takes to mount a proper offense to a particular pathogen the first time you meet it. The second time, memory cells only take 1 to 3 days to turn into their effector cells. Another reason why memory cells can mount such a much more effective response the second or third time it encounters that antigen is that the memory cells for a particular antigen outnumber the amount of naive cells that there used to be for that antigen when they're still naive. So it's about anywhere in the low end from 10 to 100-fold amount of cells waiting for that particular antigen. Another cool property about memory cells is that they're not really limited by having to congregate in the peripheral lymphoid organs in order to respond to antigen-presenting cells. They can migrate anywhere and reside in virtually any tissue, which helps them better mount an attack to subsequent reactions to that particular antigen. They're also less dependent on co-stimulators, and any antigen-presenting cell can activate memory cells. They do not have the same requirement to get activated by dendritic cells as naive cells do. Now, compared to naive T cells that get activated for the first time, memory cells tend to proliferate at a much slower rate, but their self-renewal capabilities are very similar to stem cells. So what's cool about this is that anytime a memory cell gets activated and turns into an effector cell, it also makes 2 copies, one being another memory cell and then the next being the effector cell. And this is how it both makes an army of effector cells and also maintains its memory cell pool. And has the ability to continuously do this, and we don't really see a limit on this, which is why we describe it as stem cell-like. Another cool property of memory cells is that they do not require antigen stimulation in order to survive, which is a limitation of effector T lymphocytes. Their survival, just like other lymphocytes, does however depend on certain cytokines. For instance, IL-7 is required for both CD4+ and CD8+. CD8+ also additionally requires IL-15. Memory cells that reside in the tissue and not in circulation express high amounts of the same CD69 protein that outcompetes the same S1P/R1 receptor that wants to bind to S1P in circulatory systems and wants T cells to escape the lymph nodes. So it's the same mechanism that keeps these memory cells within tissues. Now going back to effector T cells and the end of an infectious cycle, which is the decline of the T cell population. Now elimination of the antigen is really what leads to the decline of the immune response and the T cell population, which makes sense, right? As I said before, T lymphocytes require antigen stimulation in order to survive. Both antigen stimulation and the growth factor IL-2 are required for the expression of the anti-apoptotic proteins BCL2 and BCL-XL. Whenever they're deprived of either of these, then this mitochondrial pathway for apoptosis that is normally blocked by those same proteins above gets initiated, and that's what induces apoptosis. I think what a nice little recap of these last 2 notes is expressed in both of these pictures. So here we have a dendritic cell with both the co-stimulator and the antigen expressed to the T cell. The naive T cell recognizes both of these. It's dependent on both signal 1 and signal 2 in order to activate. So it does get activated. It produces the growth factor IL-2, which other T cells and the same T cell act upon, becomes effector T cells, proliferates, it differentiates, it produces both effector and memory T cells. In part B, we have an effector T cell that's already been activated previously, so it's less dependent on co-stimulators. So while it might need a little bit, it's so less significant here that it's not even expressed in the picture. So these effector T cells, they recognize that same antigen, they continue to live, They produce IL-2 that's necessary for the regulatory T cells. So in this picture, the alpha chain of the IL-2 receptor is expressed. Regulatory T cells need IL-2 from other effector T cells in order to carry on. This also continues proliferation and maintenance of the regulatory T cells, not just the effector and memory T cells. So that's it for the activation process of T cells. I think there are a lot of therapeutic applications that we talked about this, and a lot of things that we're going to get into in later chapters that are dependent on our knowledge on this. So thanks for listening. Enjoy. Bye.